This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs. Also, make sure to check out and subscribe to our YouTube original channel, UCTV Prime, available only on YouTube.
So we're going to have a quick video, so if you could just pay attention to my friend and colleague Marike.
Dr. Igor Mitrovic is a professor of physiology. He has won numerous teaching awards and is one of the most favored professors of our students here at UCSF. He gets extremely excited about the material he teaches, and it really gets you to understand the material at a whole new level. He teaches both medical students and pharmacy students, and in addition to that, he's a visiting professor at the University of Kragujevac in Serbia. So without further ado, please welcome Dr. Igor Mitrovic.
Thank you everyone and welcome. It's great to see you guys. Usually when I lecture in call hall, the situation is exactly like this. People sit far in the back, and there are these front rows that are usually empty. And it always reminds me actually of an episode from my medical school, actually second day of my medical school. There are some people here who obviously have had careers and are here for probably reasons just because they would want to have fun with some physiology, and then there's a lot of young people as well. For those young people, maybe even for those of you who feel young at heart, I just want to share this story. This was the second day of my medical school back in my former country, and I was the only person sitting in the front row. And my professor came in, and everybody was sitting in this huge lecture room way in the back. And I'm wondering what's going to happen now. And he says, guys, I can't talk to you. You're sitting too far back. We need to talk. This is lecture, of course, but I'd like to hear your voices. And people start trickling down, and there goes this woman, tall, good-looking, beautiful woman. And she walks, and now there's actually a little bit of sort of dispute what happened next. My version is that she sat 2 or 3 chairs away from me, and her version is that she sat 1 chair away from me, meaning leaving 1 empty chair. And I actually looked at her and said, and then everybody filed up next to her. And I told her, dear colleague, do I look dangerous? I'm not going to hurt you. Why didn't you sit next to me? And she smiles at me and sits next to me. It was 25 years ago, a little bit over 25 years ago. I married that woman. And I was not even hitting on her. I was just wondering what was wrong with me. Why wouldn't she sit next to me? So, you still don't want to come down? At the risk of getting married? No. Okay, well, I've tried. So I hope you can hear me well all the way back. Can you? There were some people who were saying that they may not be hearing. Well, I tend to be very loud and excited, as Marika said, about the things that I'm teaching and talking about. So I'm going to be probably very loud anyways. So you'll hopefully all hear me. So for this part of MiniMed, one little piece of physiology that was selected was cardiovascular physiology, sort of to continue to what Betty Ann was talking to you about cardiovascular anatomy. And then we were going to have Dr. Henry Sanchez talk about cardiovascular pathology. I'm going to dip a little bit in cardiovascular pathophysiology as well, but few things that I just wanted to share with you at the very beginning, and that is that physiology is science of life. It really studies how the things function. Whenever you actually think of a question, why, that is not really a very good question that science answers. Why is in domain of teleology, philosophy, or wherever you want. However, I would actually encourage you to use that question why. Because actually creating stories using question why in your head is actually the way to memorize, remember things. Our brain has evolved for storytelling. So the better story you can tell yourself about what I'm going to tell you, or anything that you're learning in your life, the better you will remember it. So that's the first thing that I wanted to tell you. Second thing is actually physiology is really concerned about a couple of themes. One of those themes is actually something that we have known about for a long time, since early 20th century, when Cannon postulated actually something called homeostasis. You probably all have heard of homeostasis. So homeostasis really means maintaining certain parameter within the optimal limits. That's actually what is happening under the normal conditions. When you actually are not functioning in homeostasis, you actually end up with the disease. Chronic diseases may be actually functioning in something that we call allostasis. Allostasis. Allo for meaning different or foreign in Greek. What does that mean? That means that you can actually end up in a situation that body actually maintains this new state that is not normal, but it's actually set it as if it's normal, set to it, which is quite an interesting thing when you think about it. For example, in cardiovascular system, we have these baroreceptors that constantly monitor our blood pressure. So because they constantly monitor it, constantly adjust it, you would actually expect that everybody always have 120/80. But we all know that there's people like myself that need to take diuretics to lower their blood pressure or some other drug. How is it then that these baroreceptors that monitor constantly can be overridden? Probably what happens is actually that as everything that we happen, and this plasticity of our body, plasticity of our brain as well, which is generally really good, enables us to actually adjust to environment, gets hijacked. So if Igor constantly gets excited by something or scared by something, and his blood pressure ends up being constantly 140/90, 140/90, baroreceptors start thinking, well, you know what? It seems that this is what he really needs. Let's shift this blood pressure and regulate him around 140/90. And there you have it. And then body regulates blood pressure around these new levels. So this is another sort of concept that I would like you actually to remember. Allostasis, that body can go there. And finally, third concept that I would like you to remember, when we talk about chronic disease, usually acute diseases are easy to comprehend because essentially something hits you and you have a problem, you can't maintain your homeostasis and you have an acute problem. Chronic diseases are associated with typically loss of physiologic reserve. Think of it this way. When we are sitting here and listening to someone like me, we don't need that much cardiac output. We don't need our heart to work that hard, unless I scare you, which I do sometimes to my students, particularly when I talk about the sympathetic nervous system. So, but if someone yells fire, which nobody will, hopefully, we're going to be rushing out and running, and we're going to have to mobilize extra resources. And those extra resources are essentially this physiologic reserve that I'm talking about. So as we're aging gracefully, hopefully, the physiologic reserve slowly decreases. In chronic disease, it actually decreases much more rapidly. So you get to the point where even at rest, you cannot maintain homeostasis. So again, another concept that I would like you to remember is essentially physiologic reserve. You've all known that people can survive with one kidney only, quite nicely, so why didn't the second kidney? Physiologic reserve. You all know that we can actually live perfectly fine, more or less, with one lung only. Why do we have another lung? Physiologic reserve. In the case of need, if there is a real need to employ something extra, we have that extra thing. And you will see that heart actually generates tons of pressure, specifically for that reason, to make sure that very quickly it can divert the blood from non-essential organs to the brain and make sure that you survive. Okay, so what are we going to be talking about? So the title of the talk is Heart Matters, Physiology of Body's Powerhouse. I actually wanted to say, little pump that could. So, when you think about the heart, this is one organ that actually has to work all of your lives. And it does work really hard all of our lives. Interesting thing is actually, if you were to put pump of the heart's kind and power to a system of tubes, metal tubes, that would be resembling completely our cardiovascular system, pump would not be able to supply the blood flow through that system. What does that tell us? What do you think? Please.
You need the other arteries and veins to support the blood pressure.
Exactly. Precisely. Thank you. What was your name?
Mark.
Mark. Thank you, Mark. So essentially, if just the power of the pump is not sufficient to provide this, that means that there is something special about the vessels or the other parts of the system that actually provides for this. Does it make sense? Yes. However, today we're going to only talk about the heart, and next time we're going to be talking about blood vessels. A couple other things that I want to point out to you is essentially that heart is, as I said, really a pump. A pump that is, it pumps, takes blood from one place, puts it in another, and it's actually controlled electronically, if you will. Strangely enough. So it's an electromechanical pump, if you will, and we're going to be talking about these two components of the heart as a pump. Electric component that is controlling its action and function, and mechanical component which is generating the force to move the blood from one place to another. Roughly speaking, because of that dualism, if you will, of the pump itself, we can talk about two kinds of problem with the pump. Electrical problems and mechanical problems. Turns out that these electrical problems always mess you up through mechanical means. But actually, if you look at where the original problem is, you can sort of separate them on electrical and mechanical problems. There's a bunch of objectives that actually we have for tonight. I'm not going to go through them. You have them in your handouts. So let's just start talking about things. So what do I start with? With the problem. Okay, this is not really a problem. This is a normal ECG recorded from a normal heart, and you can see that it looks perfectly fine. Why am I telling you this? Because actually, I want to just start with these problems, electrical problem for the start. So you can see actually this nice P wave here, and then QRS complex and T wave, and then flat line, P, QRS, T, flat line, and so on. That is how normal heart works electrically. We can record it from our extremities because essentially there is so much coordinated muscle fiber activity, electrical activity, that actually reaches up to our surface of our skin. What happens if we have some kind of a problem? First problem that I want to show you is actually atrial fibrillation. Some of you might have heard of atrial fibrillation. Some of you might have people, friends, or maybe have atrial fibrillation, which is actually a bad condition, which means essentially that we don't have this, you see this nice little P wave here? Can you find the P wave on this strip right here? Anyone? No. Not really. Indeed. And you can't find it because it's actually fibrillating. That means that there is no uniform depolarization of the atria, that the atria are just teetering like this instead of actually depolarizing to generate this current and then repolarizing, they're fluttering. It's a very dangerous condition because if you have that in your left atrium, and actually usually have in both atria, blood is not moving that much, which creates perfect conditions for generating thrombus. And if a thrombi are actually generated, from the left heart you have direct straight shot to the brain. And even small thrombus in a really bad place can actually, thromboembolus can be devastating. This is actually much more devastating condition in which you can see a little bit of the sinus rhythm. This beginning right here looks a little bit like normal heart up there, but all of these others, sometimes actually back in my med school we used to call these potatoes, because it doesn't look like anything, does it? Does that look like ECG? No, it doesn't. It actually, this is sort of ventricular rhythm when ventricles are essentially just firing however, and then the end here is ventricular fibrillation. If you have ventricular fibrillation, now that is a dire condition. This is deadly on the spot. What does that mean? Your ventricles are designed to contract, eject blood, relax so they can take more blood. Contract, eject, relax. These are just doing this. Are they pushing any blood? So what could you do for someone like this? Defibrillator, exactly. So some people would give artificial heart or do something else. The initial thing that you could do is actually just defibrillate, because we will see actually what are the things that generate conditions, electrical conditions in the heart, and actually what would be happening actually if some of those things are out of whack. Now this person actually has heart failure, which is actually a mechanical problem primarily. You may have perfectly fine electrical activity in your heart and you're still not healthy. Under certain conditions, your heart may start not be able to generate the force to eject the blood, or to generate sufficient amount of force to eject sufficient amount of blood. So what is going to be happening is, if you remember from Dr. Smoot's talk, where does the heart take blood from? From veins, and then pumps it into arteries. Right heart from systemic veins and then pulmonary arteries, left heart from pulmonary veins into systemic arteries. That makes sense, does it? So if the pump fails mechanically and can't generate the force, what do you suppose is going to happen? You're going to have some leftover blood on the side from where blood is being picked up. Would you appreciate that? And if you have leftover blood back, what do you think is going to happen? Edemas. So do you see this person has swelling in ankles and legs? Could be pulmonary edema, hard time breathing, and so on. So those are mechanical problems actually that heart actually can generate or actually suffer from. So what is a heart then? You've seen this actually. We have aorta here, pulmonary artery, left ventricle, right ventricle, and if we cut this heart, we actually have this nice little picture with left ventricle, which is actually more muscular, and right ventricle, which is much more thinner muscle. By the way, do you have any idea why the difference in the thickness of the muscle? Anybody? Guess. Yeah. Can you say it?
That it needs the strength to be able to contract?
Yes, the left ventricle actually has much more muscle because it needs to generate much more force, much more pressure to pump into systemic circulation. Right heart, nah, not really. Lungs are very compliant, very easily accept the blood, so you don't need to generate that much pressure. So what else do I want to tell you about this heart? Just these two things. One thing that I want to mention is actually that these are actually two independent pumps. Mechanically, they are independent. And because of that, you can actually have isolated failure of the right heart or isolated failure of the left heart, which is kind of a strange thing when you think. I bet you all think, well, heart is a single organ. But mechanically, they are really two different pumps, which we can see also based on the muscle that actually is found in left ventricle versus right ventricle. Another thing that I would like you to remember about the heart is that the heart, there are different kinds of pumps. Heart is actually a pulsatile pump. What does that mean that it is a pulsatile pump? That it doesn't provide constant pumping. It actually pumps, then rests, pumps, then rests, and hence we have actually these sounds, heart sounds, and two actually cycles. If you will, it's two-cycle machine. Systole and diastole. Systole is when it contracts, diastole is when it rests, when it relaxes, and it relaxes and it rests because it actually needs to have very low pressure to accept the venous blood that is coming at a very low pressure. Therefore, it has these tricuspid and mitral valves, which are essentially, once it contracts, preventing blood going back into atria and the veins, because that would be actually the place of the least pressure and the easiest place to go for the blood if it wasn't for the valves. So this is what the heart does then. It pumps into systemic circulation and pulmonary circulation. Systemic circulation generally starts with left heart, it ends with left ventricle, and ends with right atrium. So essentially from here all the way to here is systemic circulation. One thing that I want to tell you is actually, if you take a closer look here, maximum pressure that our healthy heart actually is generating typically is around 120 millimeters of mercury. And the pressure in a right ventricle, healthy pressures are around 2 millimeters of mercury. You see that? This is a huge pressure drop. And I'm calling it on purpose pressure drop. Anybody knows what other name I could have used for it? A driving pressure. We could call that a driving pressure. Difference between the 120 and 2. So that's what is happening on systemic side. On the pulmonary side, actually, I said already to you that right heart actually really doesn't need to generate much pressure. 25, and this ends up around 5 or 4 or thereabouts. Why is that? So can anybody guess why is it that left heart actually has to generate so much more pressure than the right heart? Yes?
Traveling a lot farther, and it's got to get the blood all the way to the feet and head.
Much longer pathway, indeed, there's much, much longer. So if we actually take this length and convert it into some kind of physical unit, what would we call that? What would that be contributing to? Length. Someone said it? Resistance, indeed. So essentially, the reason why left heart is pumping so much and generating so much pressure is to overcome large resistance. There's a lot more resistance to blood flow in systemic circulation than there is in the lungs. So generally, that driving pressure is actually there to essentially overcome the resistance. Which means that if, say, we have a problem and have aortic stenosis here, sometimes that happens in certain diseases. You may have some rheumatic fever and then valve gets damaged, or someone is born with actually very narrow aortic orifice. And you have a huge resistance here. That heart, under those conditions, may actually have to go not 120, but 180. And yet you measure arterial pressure, and arterial pressure may be 120/80. All that extra force is used just to overcome the resistance at that narrow valve. So does this make sense, guys? So heart generates the force in order to overcome the resistance in the system. Now, that's actually sort of this big picture. Now we're going to get a little bit into details and start talking a little bit about the electrical system in the heart. Heart cells are a little bit like batteries, and actually it's billions of years of old batteries. How are they like batteries? Well, think of it this way. I have actually here represented single cardiac myocyte, and there is a situation in cardiac myocyte that you have much more positive ions outside than inside, turns out, within the membrane. If I were to stick an electrode here in the living cardiac myocyte, I would be able to measure maybe minus 80 millivolts difference between outside and inside, outside being actually reference 0. How do we end up with that? Well, turns out that we have some pumps. You probably have heard about these specialized proteins. Some are actually actively using ATP to pump potassium inside of the cell and sodium outside of the cell. Calcium is a very dangerous and extremely important molecule. It can wreak havoc if it's left unattended in the cytoplasm. That's why heart actually stores it in sarcoplasmatic reticulum. And you can see that inside of the cell, free calcium concentration is extremely low. And it's kept low by these different pumps. One is actually sodium-calcium exchanger. And then there's actually calcium ATPase, which uses ATP to pump calcium out and calcium in the plasmatic reticulum. So this is what sets these concentrations like this. We still didn't generate these negative charges inside of the cell relative to outside. There's another set of proteins that actually do that. And those are voltage-gated ion channels. These are specialized proteins that are sensitive to voltage. They actually sense when the battery changes its voltage and essentially let only certain types of ions out. We have something called leak potassium channel that is sitting right here, and because potassium is very high inside and low outside, potassium constantly leaks out. And because potassium is just about the only, I'm simplifying things a little bit, it's not the only, but just about the only ion that is allowed at rest to leak out. What we end up with is actually a bunch of positive charges lining up around the outside of the cells and negative charges inside of the cell. And that is actually what generates this battery that I was talking about. Do you see that? Now, you were going to ask me, Igor, you seem to be teasing us because you didn't, how can this be negative? You didn't write in, there are no negative ions in here. There are no anions. What we need to remember that each of these cations actually has its attended anion at the same time. So essentially you have anions here and anions here, so negatively charged. So I just didn't draw them for simplicity reasons. But you will appreciate the fact that if we let something positive leak out, that inside is going to become more negative and outside is going to become more positive. Does that make sense? And there's a battery. And what if we actually turn that battery on? Can we turn the battery on? Look at this. This is our ace in the hole, sodium. If we could somehow make this membrane permeable to sodium all of a sudden, that sodium could rush in like mad because inside is negative and there's tons of positive sodium outside. Do you see that? And that's indeed what happens. That's how we turn this battery on. And all excitable cells in our body work like that. Muscles in my biceps, hopefully neurons in my brain, and the heart muscle cells and conductive cells in my heart. So what I'm going to show you here is actually a specific turning on of the heart muscle cell. This is called an action potential, and this is how it happens. This is time, and we have membrane potential change. Cell is resting at very low negative potential because of this leak of the potassium that we've talked about. And then all of a sudden there comes a little signal from a specific part of the heart called SA node that sends these impulses regularly, which is a pacemaker, and it depolarizes the cell. And look what happens. At this point, voltage-gated sodium channels open and sodium just rushes in, moves the cell into more positive side, and then there's this tug of war between calcium and potassium here, and then sodium channels inactivate, calcium channels inactivate, and potassium now leaks big time out just to return the cell back to normal. But what we got is a signal. I'm not going to go into particulars of these ions and of these channels. Just remember that sodium is the carrier of this signal. And it's carrying this signal because actually there's a lot of sodium outside, huge concentration of sodium outside, and cell is negatively charged on the inside. So if you open voltage-gated sodium channel, sodium is going to rush in and create this. Very important point that happens there at the same point is actually also that calcium is allowed to go in, because as the membrane potential changes, these channels are voltage gated, which means essentially voltage turns them on and off. And that calcium is going to become important later. So take a look at what happens then with the heart. If we have actually heart and cut out piece of the heart, we have these heart cells. This is just simple histology. We have intercalated discs. And if we magnify one of these intercalated discs and take a look how that looks out, we have special connections between the two heart cells. We have these desmosomes, which are extremely, extremely strong connections between the heart muscle cells. Why would you want to have extremely strong connections between the heart muscle cells, guys? What do you think? Say it.
So you don't blow it apart.
So you don't blow it apart, because essentially each heart cell contracts on its own. So you have to tighten them very tightly so that once they start contracting, they just start trying to pull apart from each other. So if you don't allow them to pull apart from each other, what happens? Can you two guys come outside for a moment, please? And someone else. Can you come? We need to be here within the camera so that in the light. So let's give our hands to each other. So we are 4 heart muscle cells. So if I ask you to contract your biceps and do this, without touching each other, so or just lightly holding each other, contract. Heart rips apart. Now do the same thing and hold on tight. See what happens. If there was some fluid in between us, we would be squeezing that fluid, and that fluid would be going. Thanks a lot. Thank you, guys. So that's why desmosomes. And then there are these other things called gap junctions. What are the gap junctions? Gap junctions are actually holes. Physical holes, protein-made holes, that allow sodium from this cell to jump through the gap junction to flow into this cell. Why? Remember that signal that arrived when we turned it on? When a lot of sodium rushed into this cell became very more positive and it had a lot more sodium than this cell. And that extra sodium now concentration is greater here than here. And that sodium comes in the next cell. And that depolarizes that next cell, which opens more voltage-gated sodium channel, which allows sodium from outside to rush in. And then, boom, another signal. And the next cell, boom, another signal. And you essentially have spread of action potentials from one to the next, to the next, to the next cell. And you have essentially created movement of electricity through your heart. Beautifully designed system. It has to be done really, really in a coordinated way. And this is actually just the shadow of the heart. I believe Dr. Smoot actually told you that everything starts from the atria. There's this SA node, typically in a healthy person, somewhere around the vena cava superior, where's these pacemaker cells, which are really unstable. And I'm not, okay, let's not go make jokes like that, but they are really unstable. Their membrane potential is not flat. They actually go up and down, and they're called pacemakers because of that instability of their membrane potential. They actually generate constantly these signals that keep repeating. So signal starts here and then actually depolarizes all these atria. So this is SA node. And here, between the atria and ventricles, there is another node called AV node. AV stands for atrioventricular node. And that one is actually there to slow down a little bit the conduction between the atria and ventricles, because what you want is actually your atria to contract before ventricles. And then we have a bundle of His. And then bundle branches, right and left bundle branch, and then Purkinje's finally. And what I'm actually showing you here is a conductive apparatus. These cells that I drawn in yellow, left bundle branch and right bundle branch, are not really a muscle. They're modified muscle cells that are there really to conduct electricity, to make sure that electric impulses are spreading in a certain direction with a certain speed so that the timing is right. So timing electrically in the heart is everything. If you mess up electrical timing, you end up with that stuff that we've seen, those fibrillations. And a lot of things actually can cause them. Keep this in mind, because I'm going to actually ask you a few things. Yes, please.
Why are the atria and the ventricles seem to have different structures for transmitting the signal? You don't get those bundle fibers apparently up in the atria?
A simple reason is probably, this is a question why. You know that we are now in the teleology area.
Operate mechanically or electrically, there must be some difference in the way they operate.
So there isn't that really a big difference. There are some fibers, actually specialized fibers, even in atria. It's just that we don't care that much about them. There are specialized fibers that are conducting from SA node to AV node, probably. But actually spread, even in the ventricule, is actually, once you get here, is from cell to cell. And it's actually fast enough. The thing with the ventricule is actually that you want the tip of the heart to start contracting first. And in atria, it doesn't matter, because actually the contraction starts from the top and it's squeezing towards the ventricles. Does it make sense? And in ventricle, you don't want the septum to start contracting or this side of the heart to start contracting, because you know where the major arteries are coming off. They're coming off at the base. So you want tip to contract first.
That's the reason, okay. So was it the top, was it the bottom?
Exactly. Thank you. Thank you for the question. So please, if you actually have any questions, stop me. I can stay here for a number of hours. My wife usually doesn't expect me home when I'm teaching. Okay, I'm just kidding, obviously. So what I wanted to show you here is actually those action potentials that I've illustrated for cardiac myocyte. This is the one that I actually have shown you. Depending where in the system we are, we actually have different shape of action potentials. So if you take a look at up here, SA node has this. Do you see how this thing actually dips down and then slowly drifts up? And this isoelectric line is -80, is just staying flat on the ventricular myocyte. So you can appreciate that this action potential and this action potential are different from the rest of them. And those two are primary pacemakers. SA node is primary pacemaker for the healthy heart. AV node can become a pacemaker in atrioventricular block. If the AV node doesn't get the signal from the atria to say contract now, it will start giving a heart pace. This one actually works at the pace of approximately 70, as you would expect. In my heart, it's more like 48, 50. This one, it works around 48, 50. Curiously enough, all heart muscle cells can become pacemakers, which is actually a danger in its own right, as you can imagine. If you mess up electrolytes, those potassium, sodium, and so on, you can actually create problems. What would happen if we would have a signal that starts here? You can imagine that could represent a problem, could you? Or signal that starts here. Obviously, it wouldn't be spreading normally. Turns out that these pacemakers are actually controlled, because you want to be able to control how fast the heart is working. And you're all quite familiar with that, probably. If I do something like crazy, which I'm not going to do here, and yell, your heart rate would probably go up. And it would go up because I've scared you. And sympathetic nervous system would be actually stimulating your pacemaker, making it fire faster. And if you actually are really bored by my lecture right now, and it's kind of late, you might have had a dinner, and you're kind of resting and want to kind of relax and maybe fall asleep, your parasympathetic system is going to be active. And it will actually inhibit the pacemaker, make your heart pump slower. Those two systems are obviously used for adjusting the needs of the body. You can change how much blood is pumped by changing how fast heart works. We will see in the moment that actually sympathetic nervous system can also increase the strength of the contraction of the heart muscle. Curiously enough, this pacemaker is also regulated by SANS and PANS. So pacemakers actually have this, as I said, oscillating electrical potentials. And actually they have less ion channels than ventricular muscles. Well, I shouldn't say that. This is not entirely true. They have different complement of ion channels. So this would be actually how they don't have isoelectric line. And you can see they are not sitting there at -80. They're much more positive under normal conditions. So they have this. The reason why actually this is going this way, this membrane potential keeps depolarizing, is particular. There is a fun story around this. It's called IF, or funny current, funny channel. It's a sodium, mostly sodium conducting current channel. In those other cells that I've shown you, these voltage-gated sodium channels, when they are depolarized, sodium comes in, channel opens. This one actually is exactly the opposite. This channel opens when membrane is repolarized, when the membrane becomes more negative, and it passes sodium. So there is, I keep forgetting the name of a British gentleman who actually discovered this channel, who actually looked at this and saw it was a sodium channel. And it was unlike any other sodium channel. Normally voltage-gated sodium channels open when membrane is depolarized. This one opens when membrane is repolarized. Exactly the opposite. And he was looking at the data that he was recording and said, "Oh, how funny." And that's actually funny channel. Indeed, IF actually F stands for funny. So then calcium channels open, and then calcium, and you see what happens. Pacemaker constantly paces, goes up and down, up and down, up and down. And whenever action potential occurs, it spreads through atria, goes down to AV node, bundle of His, bundle branches, Purkinje's, ventricules. Next one, same thing. What our sympathetic nervous system does, I'm going to use red for sympathetic. Hopefully you can see it. Nah, I'm going to use green for sympathetic because it makes it faster. So sympathetic system does this. Essentially makes it occur much more rapidly in time. Parasympathetic system actually does the opposite. Instead of actually this happening like this, it says, "Oh no, you're not going to get there to fire. Now you're going to fire a little bit, and then we're going to get down." So that's what sympathetic and parasympathetic system do. Does it make sense? So this is PANS and this is SANS. Atrial cells are turned on before signal arrives to ventricular cells. Yes?
So are you saying that the yellow system is sort of the sum of those two?
Actually, you could say that. Because typically in a healthy heart we have essentially tug and pull. There's actually quite an interesting device that I use from time to time to calm myself down when I get too excited about teaching. M-Wave is HeartMath is actually the company that makes device. And it's essentially based on the fact that you have oscillation of parasympathetic and sympathetic system that are essentially regulating heart rate. And when presumably someone is in good coherence, when one is really relaxed, then you could even see those oscillations in the heart rate, two types of oscillations. You can see actually these slower oscillations of parasympathetic nervous system and faster oscillations of the sympathetic nervous system. If you actually plot heart rate, or just heart rate dots over time. But you're right, exactly. These two systems are actually kind of playing off of each other. Although in most of us, at rest, parasympathetic system predominates. If I were to do surgery on someone of you and remove parasympathetic innervation of the heart, your heart rate would go up to 80. But a great question. So atrial cells are turned on before signal arrives to ventricular cells. And the reason is because you want atria to contract before the ventricles. That makes sense, does it? And you want atria to actually kind of be this extra push of blood in the ventricle before ventricle starts contracting. Now, this is not a lot of blood. This is 5 to 10% of the total blood. So you don't really need this extra blood. If you were actually some kind of Olympic swimmer or runner, that would be actually making a major difference. But in an average person, that extra pump is not that important. But just to illustrate to you how this happens, this is how it happens in time. So somewhere here we have had our SA node trigger this, and we have action potential in the atrium and action potential in the ventricle. Does it make sense? So they're timed. First atrium, then ventricle. Why is that important? Because essentially, as I've shown you before, we want contraction to occur right here first before it occurs here. Curiously enough, that same thing can be registered actually on ECG. So what I've represented here to you are just two cells. One atrial and the other one is ventricular muscle cell. Only two cells. However, what happens is that you're going to have a mass of atrial cells that are depolarizing approximately at a similar time. So you're going to have a bunch of cells going around here. And you see, because it's happening at the same time, this depolarization, we're going to actually see that as because it's a large mass of atrial cells, we're going to see that in ECG as a P wave. P wave is atrial depolarization. Then we're going to have this isoelectric line, which, you know, there is not much change. When there is no change, when the current is not traveling through the heart, you don't see really anything in ECG. ECG is generated only when the current is traveling, when you see the differences between the two arms or arm and leg in electrical voltage. Then what happens is actually our ventricle starts contracting, and then you have even more, much more ventricular cells that are actually depolarizing, tons of them, tons of sodium going from out into the cell, from out and into the cell. And it happens so that actually mostly left side depolarizes because there's much more muscle there, a little bit slower than the right side, so we essentially end up with something like this, QRS complex. QRS complex, this is Q wave, R wave, and S wave, is actually due to ventricular depolarization. So when ventricles depolarize, this mass of cells, we see this. Does it make sense? And then, because we have this flat line here, no, doesn't matter that the flat line is not that, in these negative sort of, it's not here. What matters is that there is no change. Nothing changes until ventricle starts repolarizing. This starts going down. And it goes down and down and down, and all of the cells of the ventricle start repolarizing, and they're repolarizing kind of more or less within certain specific time period. And then we get this T wave, which is actually ventricular repolarization. So this is how we get our ECG. We can see that, measure it actually very easily from the surface, because there is a mass of current that goes in one direction. And you can also mess it up if you actually put ECG on someone's arm and then they actually play with you and contract their biceps. Then it doesn't look like this, it looks more like this. So the person has to be relaxed, obviously, when they're doing this, because all of our muscles are actually electrically conducting. The beautiful thing about ECG actually is that this was the first time that one could actually record something that is actual activity in a living body, living human body. And Eindhoven did it at the turn of the 20th century. And in essence, nothing really changed from the time that he actually did it. We actually discovered that we could also put some electrodes in front of the heart so that we get sort of different view of the heart. But in essence, didn't really change. It's more than 100 years old. He got Nobel Prize for that. And where the electrodes are actually kind of is curious, because where you position those electrodes actually dictates what part of the heart you see better. So these electrodes, they're typically the Eindhoven triangle, right arm, and left leg. It gives you actually cross-section, this coronal plane. And these precordial ones cut it this way. And if you actually have that, if you have essentially done that, you have defined two planes. And what are the two planes defining? Three-dimensional space. So you can really, by analyzing carefully which electrode lead or which electrodes are recording, you can actually be very precise in determining if someone has had a myocardial attack, which part of the heart is actually affected. This is a remarkably beautiful system. So, this is that normal ECG that I've just talked about. P wave, QRS, and T wave. And then a pause for the heart to receive the blood. And then P wave, QRS, T wave, and the pause for the heart to receive the blood and fill. Now, why are these strange things like fibrillations occurring? What can mess up electrical conduction in the heart? What do you think? I will actually leave you a minute or two just to think a little bit about it amongst yourselves. What could actually cause the heart to go from here to atrial fibrillation like this? Or ventricular fibrillation like that?
Would the electrical system be affected by mechanical blockages like cholesterol, like clogged arteries?
It could. It could, if you actually, but then you would have myocardial infarction. And you wouldn't be worrying about this. Say it, please. Ion channels? Who said ion channels? Raise your hand, please. What do you mean by ion channels?
Sodium or sodium ion channel that's gated to be off.
indeed, there are some genetic problems in some people who actually can have particular variants of potassium, high potassium channels, or particular genetic variants of sodium channels that actually cause something called long QT syndrome, which can actually prolong this particular thing. And actually, if you prolong something in heart, you can create a situation in which the heart is actually starting to discoordinate. And you may actually have situation in which next impulse is actually generated within the tissue before it actually arrives from SA node. So indeed, problem with ion channels. What else? Yes?
A lightning strike?
Lightning strike. I didn't think of that one, but but lightning strike could do that. Actually, yes, exactly. I've had pretty bad situation with my brother who was playing a guitar with the amplifier that wasn't grounded and actually touched the microphone and fell down and actually was shocked really severely. Yes, that can be something, but let's stay within the body. Yes.
So imbalances with sodium, calcium.
Excellent. Imbalances with electrolytes. They, particularly potassium in particular, because it's actually low outside. So changing it from 4.5 or 4, which is normal, to 6 or 7 can actually really change what is happening with these things. It turns out that there are even some ion channels that change what they're passed through if you mess up with potassium a lot. Excellent. Yes?
So if you have like dehydration, can that in fact cause the imbalance?
Dehydration can cause the imbalance, in particular if you dehydrate and then you drink water. That'll cause actually something else, bigger problem in the brain rather than in, but yes, you can. Than in the heart, yes. So those are all the things that can do it. Small myocardial infarction indeed also can cause a block. If you actually block bundle of His, so essentially it's as if you transected actually this connection, electrical connection, and if you transect the electrical connection, that you're left ventricles to do their own thing and then atria are doing their own thing. What would you do in that situation if you have this condition, atrioventricular block, where essentially atria are doing whatever they want to do and ventricle is actually doing it but very slowly, because ventricles don't actually, even though they can pace on their own, they pace very slowly. What would you do? Yes? Pacemaker, indeed. You're not a medical student, are you? Okay.
I watch a lot of House.
Oh, you watch a lot of ER, okay. So pacemakers, you can use essentially some of these external devices essentially to pace the heart properly. This is what I was talking about, the blocks. If you see normal sinus rhythm and you see, do you see these P waves? This is one, this is another. Do you see how they don't actually really correspond to QRS as they normally correspond to QRS here? Each P is followed by QRS on this side with the normal rhythm. On this side, P does this its own thing and QRS does. Another thing that you can see on this QRS, can you? Does it look funky? Doesn't look quite this spiky QRS, does it? Yeah, the reason is because the actually spread of the current is not as appropriate as it should be. Obviously, this signal is originating somewhere else, not coming from AV node. So essentially, you don't have this normal spread in left and right bundle branch. There's probably some branch block as well here. So that essentially you have spread of the current that is actually going in a round and about way. So QRS is wider. It takes longer time for the heart to depolarize. So what did we say thus far in a little pump that could? We've learned that heart actually electrical activity is controlled by specialized conductive pacemaker and conductive cells. We actually said that the electrical activity can be regulated by sympathetic and parasympathetic system. we actually talked about electrical activation of the heart muscle and how it generates ECG waves. And then we said that irregular electrical activity results in heart problems. These irregular heart activities obviously are going to create biggest problems mechanically, because if you actually mess up with normal conduction, your heart is not going to be contracting properly, not generate the current properly. So, but how does the heart work as a pump? How is this little pump that could essentially doing what it does? Well, there are a few things that I want to show you. First thing is actually this diagram that actually shows you quite an interesting thing. These here are myofibrils. These are contractile parts of the heart muscle. And myofibrils have actually these calcium sensors, troponin C, where calcium binds. And calcium is the most important thing for contraction of the heart muscle. As a matter of fact, concentration of the free calcium in the cytoplasm is what determines the strength of the contraction of the heart muscle. That's why calcium concentration is extremely tightly regulated. One other thing that I want to show you is actually that these cells have these transverse tubules. Do you see this thing here? These transverse tubules are invagination of the membrane, cell membrane, that goes deep inside so that it can be close to each myofibril and can be really close to sarcoplasmatic reticulum. And sarcoplasmatic reticulum is essentially calcium storage place. So there is tons of calcium sitting here, waiting for action potential to come down this membrane and to actually trigger the reaction. And what happens is essentially when action potential comes, travels down, depolarizes this membrane, and this is L-type calcium channel. When the membrane depolarizes, it opens, this is voltage-gated channel, it opens calcium channel, L-type calcium channel, calcium rushes through, and actually opens up this ryanodine receptor that is sitting on a sarcoplasmatic reticulum, which then allows tons of calcium, which is actually bound very loosely to something called calsequestrin. You don't need to know that. Well, do you really need to know any of this? But it's fun, isn't it? It is a lot of fun. So calcium rushes out and then gets down to these contractile fibers and enables them to contract. So what I'm going to show you now is a little movie on how this happens.
This animation sequence will provide an overview of normal cardiac structure and physiology, along with a review of the process of excitation-contraction coupling in the heart. The myocardium is comprised of cardiac myocyte and non-myocyte cells. Cardiac myocytes make up approximately 70% of the mass of the heart and are enmeshed in a collagen network that acts as a supporting framework. Blood is supplied to individual cardiac myocytes through capillaries that are in close proximity to them. Cardiac myocytes are comprised of actin and myosin myofilaments, whose organization gives rise to the characteristic striated appearance of cardiac muscle. There are two prominent organelles within cardiac myocytes: the mitochondria, which produce energy, and the sarcoplasmic reticulum, which regulates calcium handling in the cell. Cardiac contraction is initiated when extracellular calcium moves into the cell across the voltage-dependent L-type calcium channel. The calcium that enters the cell triggers calcium release from the ryanodine receptor through a process known as calcium-triggered calcium release. Calcium that is released from the sarcoplasmic reticulum binds to the actin-thin filaments, thereby initiating contraction of the sarcomeres. At the end of the contraction sequence, calcium comes off of the myofilaments and is actively transported back into the sarcoplasmic reticulum by the calcium pump in its membrane. Calcium is also transported out of the cell into the extracellular space by a membrane-bound pump sodium-calcium exchanger, which is not shown here. Cardiac contraction is brought about by interactions between the actin-thin filament, shown in yellow and purple, and the myosin cross bridges that project from the thick filament, shown in red. In relaxed cardiac muscle, the conformation of troponin and tropomyosin prevents actin from interacting with the myosin cross bridges. As a result, actin is unable to interact with the myosin cross bridges containing bound ATP. In activated cardiac muscle, calcium binds to troponin C, which then shifts troponin complexes and tropomyosin to an active conformation that enables actin to interact with the myosin cross bridges. Myosin ATPase on the myosin heads then hydrolyzes ATP, thus providing the requisite energy for the myosin cross bridge to interact with the thin filaments and pull the thin filaments towards the center of the sarcomere in a ratchet-like fashion. In this sequence of the normal heart, we have reviewed the process of excitation-contraction coupling. As will be discussed subsequently, this process of excitation-contraction coupling becomes dysregulated in cardiac hypertrophy and cardiac failure.
So, this is what I wanted to show you actually. This is beautiful video. Don't you think that this is actually really beautiful? To think that this is happening as we are sitting right in all of us right now. These gazillions, Google number of cross bridges forming all the time while we are just sitting here and enjoying ourselves learning. All Things Science is, by the way, very nice site if you want to use that site. There's actually quite a nice YouTube videos to check out. So what makes fluid move through a tube or a set of connected tubes? Just a little reminder of physics. You will probably remember that fluid moves only if there is a pressure difference. We agree with that? So if the pressure here, oh, I need my pen. Is much greater than P2, then the fluid is going to move. This pressure difference is delta P. We already actually said that flow is actually proportional to this pressure difference. And we already ascertained that the reason why we need to generate this great pressure difference is resistance in the system. So that's why that essentially what heart does. It generates the pressure in order to move the blood. It generates it. We've talked about this systemic circulation from here to here, and much smaller pressure from here to here. This is just actually where that all blood goes if you actually want to take a look at it. You don't have this slide. You don't really need to have it, but this is just an illustration. Small, if you take a closer look at actually, you will see that pulmonary circulation is much, much, much smaller than systemic circulation. And then in systemic circulation, we have a bunch of these parallel capillary beds that go to different organs as well. We're going to talk much more about blood vessels next time. So what I wanted to show you now is actually what really happens with the heart as a pulsatile pump. So the red line here is actually showing changes in pressure in the left ventricle. Left ventricular pressure. This thing here is aortic pressure. So what am I trying to show you here? If you take a closer look, you will see that pressure in the left ventricle is going from very low right here. And do you see this tiny black line above? That's the pressure in the left atrium. You see how the pressure in the left ventricle has to be below the pressure in the left atrium in order to blood to go from the left atrium into left ventricle. Does it make sense? And then we have our QRS, which is essentially contraction, causing contraction of the ventricle. And ventricle starts contracting, and you see what happens. Pressure in the ventricle overcomes the pressure in the atrium. And that is the point when you hear first heart sound. AV valve closes. And we have some point, to some point if you take a closer look, ventricle is contracting, but pressure in the aorta, which is right here, is still much higher or higher than pressure in the ventricle until the ventricular pressure overcomes the pressure in the aorta, and then blood starts leaving the ventricle and pressure slowly drops. You can see this is the emptying of the ventricle, volume of the blood in the ventricle dropping down. Does it make sense? And at some point, ventricle reaches the maximum contraction, and then it starts relaxing. Starts relaxing because those pumps have pumped calcium back into sarcoplasmatic reticulum. Calcium is not sitting on troponin C anymore, so essentially cross bridges are not forming. And you have relaxation of the ventricle. So you see how the ventricular pressure starts going down. And once ventricular pressure goes below the aortic pressure, you have second hard sound, when it's essentially aortic valve slapping closed. Does it make sense? And that's those lub-dub, lub-dub, lub-dub sounds. Lub is atrioventricular valves, both of them, and dub is pulmonic or aortic valve. Makes sense, does it? So in order for ventricle to actually fill again, pressure has to drop below the pressure in the atrium. And you can see how slowly blood keeps coming in the atrium, because blood from the lungs keeps coming in the left atrium, keeps coming and coming. The left atrium is kind of compliant, and it's actually allowing it to come in without changing much pressure. But pressure is slowly rising even in the right atrium, left atrium. Do you see that? The black line is going slowly up, and the ventricle is relaxing, and at some point you have essentially crossover. Pressure in the left ventricle drops below the pressure in the left atrium, mitral valve opens and blood rushes in the left ventricle. And we have essentially, again, filling, and the next cycle is going to start. When does that relaxation starts? Well, with the T wave. Does that make sense, guys? So that is essentially the same thing happens on the right side, it's just that pressures are much smaller. Pressure gradient is just going around 25, and you can see that this one is actually going up to 120. So I actually wanted to quickly just ask you, what do you think? Where do we measure systolic pressure in the ventricle? At what point would you say, well, this is the systolic pressure? At the very tip, very top, exactly, right here. So let me just change to felt tip pen. So this is the systolic pressure. You can see that there's actually very little difference between ventricular and aortic systolic pressure. If there were aortic stenosis, ventricular pressure would not look like this, it would look like this. And aortic pressure would still look the same. Where do you think we measure diastolic pressure in ventricle? All the way down here, somewhere around here, wherever it's the lowest. What about in arteries? Diastolic pressure in arteries. Can you point on this graph, where would that be measured? Right here, just before of the opening of aortic valve. That is the lowest point that the pressure is going to be in the arteries. As soon as it opens, you are jamming new blood from the ventricle in. And it actually, as you can see, slowly, once the ventricle actually, aortic valve closes, pressure slowly keeps dropping, 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 dropping until it reaches this point again, and so on. Does that make sense? By the way, is anybody trying to play the game? How many times did I say, does it make sense? I just noticed I say it a lot. So this is actually how sympathetic nervous system can increase the strength of the contraction of the ventricular contraction. It actually works. This is going back to your question, who asked about specific molecules? You did, yeah. So norepinephrine released from the nerve terminal here binds to beta-1 adrenergic receptor, activates particular G protein, which you don't need to know, and actually activates the adenylyl cyclase. And then the activated adenylyl cyclase actually activates cyclic AMP, and cyclic AMP activates protein kinase A. You don't need to really know this. I just wanted to show you that phosphorylation is the thing that is actually the key thing. So this protein kinase A is going to phosphorylate this L-type calcium channel and make it more likely to open and stay more open. And if it does that, what's going to happen with calcium? More calcium comes in and releases more calcium from sarcoplasmatic reticulum, which is then going to enable more cross bridges formation and much stronger contraction. Does it make sense? So that's how sympathetic nervous system does it. It's beautiful, isn't it? Yes?
Why doesn't the calcium directly trigger the
Why doesn't calcium from L-type calcium channel do this? Some of it does, but there isn't that much calcium that comes from through L-type calcium channel. Some of it comes from L-type calcium channel, but there is tons more in the sarcoplasmatic reticulum. It's like a little reservoir that is actually waiting, just waiting. Is it coming? Is it coming? Is it coming? So what happens actually if the heart fails to generate enough force, and that means enough pressure? We can obviously, I've just shown you, regulate how much force and pressure heart is going to actually generate in pump. There are other things actually that generate how much cardiac output you end up with. You can actually play with the veins and actually increase the venous return. Sympathetic nervous system can actually make veins tighter. We're going to talk about it next time. But what happens if the heart doesn't generate enough force? You can die. You can die, indeed. That's actually the ultimate. But before ultimate, we're going to have stasis of the blood behind. And if you take a look at these areas, if I told you now, my left heart, which is right here, is failing. You can actually have left heart failure independent of right heart may be fine. What do you suspect would be happening if the left heart were failing? Take a look at this picture. So not much blood would be pumped from the left atrium, and that would cause what?
Trouble breathing.
Trouble breathing, indeed. So you would actually have edema in the lungs. So if you've heard of people who actually have heart trouble and they need actually these extra pillows to sleep, they need them essentially to keep upright, because when they lie down, edema gets worse. This is due to left heart failure. They also cough more.
So that's congestive heart.
That's congestive heart failure. Congestive heart failure. What if my right heart failed? And actually, typically, it turns out right heart fails more often than the left heart. What would happen if we have a right heart failure? Take a look at what that is going to do. Swollen ankles, peripheral edema. Instead of lung edema, you would have peripheral edema. You could actually, under certain conditions, actually have also, look what is the organ that is really close to the heart and right next underneath the diaphragm. Which organ is that? Liver. So liver, you can actually damage liver as well. And because there's tons of blood vessels in your abdomen, you can actually have essentially ascites, swelling of the abdomen from all of these blood vessels filtering into your abdomen, and you essentially have this situation. Ascites, swelling in ankles, typically right heart failure, coughing, pulmonary edema, left heart failure. In both situations, you would also have severe problems in fitness, obviously. Depending on how bad your failure is, you could actually be able to climb 3 flights of stairs only, or 2 flights of stairs, or you could just sit. All of this is just mechanical. Your electrical could be perfectly fine. And the last thing is, what could cause this? What could cause actually heart failure? Congestive heart failure or chronic heart failure? What do you think? What was that? That is much more rare to actually be born with it. But I suppose that's a possibility, yes.
Blockages in the coronary circulation?
Blockages in the coronary circulation are more commonly going to cause myocardial infarction, and that can lead then to chronic heart failure. Indeed.
Well, at the beginning you were talking about we have to be in homeostasis, and if chronically we have a problem, then it changes the balance.
You are absolutely right. And one change of that balance is high blood pressure. Uncontrolled high blood pressure causes heart failure, because it asks the heart to do extra work all the time. Does anybody watches Formula 1 races? Ever seen of those? Or IndyCar races? Formula 1 race cars actually have these engines that are actually designed to run at very, very high RPMs, 18,000. Does anybody here have Toyota Corolla? It runs forever, doesn't it? Almost. So Toyota Corolla, 200,000 miles. Formula 1, 500 miles. The one race, essentially. So the thing is, if you ask heart to do extra work, if you ask it to do much more than it's actually typically should be doing, it is actually going to hypertrophy, and then hypertrophy actually can lead to heart failure, because heart can't perfuse itself properly when it's hypertrophying. And you can have replacement of these normal heart cells with, scar tissue, and essentially scar tissue is not normal muscle. And then you have heart becomes flabby and dilated, and you end up with that heart failure. So anything that requires heart to do extra work can cause heart failure. So can do certain viruses, some bacterial diseases, alcoholism. All of these things can actually cause heart failure. So I hope I didn't cause heart failure for you tonight. I've had great time. I'll stay for anybody who wants to ask me anything else. And thanks a lot for your attention. It's been a pleasure and honor to be here with you tonight.